There's a lot of different ways to go here. I'll focus on three. We'll keep it at that. There's more that we talk about in the paper, but the first one that I would suggest is one of the goals for the next 10 years really has to be defining a metric for what makes a well person, a healthy person, well. Right. So if we brought in 1,000 different people right now and we did this deep phenotyping and we took all of these measurements for these otherwise healthy people, we're going to get 1,000 different answers for what is healthy because we're all unique. A phrase I've heard is that healthcare is one of the only industries that doesn't have its own gold standard. When we go into the doctor, we're compared against population averages because I don't have a gold standard for what does it mean for me to be healthy or for you to be healthy. So that's one of the absolute key points that we really need to focus on is how do we quantify wellness? One of the ways to do this perhaps comes down to this concept of defining your biological age. So your chronological age is, how many birthdays have you celebrated? Your biological age is a developing research field that attempts to understand all of these different modalities in your body. How do they contribute to how your body functions? There's been some fantastic work, again, out of ISB here with John Earls and Nathan Price and Lee Hood asking these exact questions and trying to understand how does your biological age, how can that be used to quantify how well you are? And again, it's important because if we're able to quantify our wellness, then we're also able to quantify when we're not well. And this goes back to this N of 1 idea, where if I have my baseline and I understand what my wellness is, I can start to detect deviations and as we move away from there. The second point that I would discuss is it's going to be important moving forward, and particularly to your last question about the affordability of these kinds of technologies and this kind of approach, is to identify those individuals who are at high risk for developing different cancers. So if we're able, potentially from the genome or maybe from the genome and some other measurements, to identify whether an individual is at higher risk for developing a brain tumor, for example, then we can have that individual go in and start getting an annual MRI, for example, whereas giving every single person an MRI annually is certainly not feasible on many levels. But if we're able to do that for a targeted group of individuals, then we could really improve the chances of diagnosing these kinds of diseases. And ultimately, that's really what it comes down to, is diagnosing these kinds of diseases, these state changes, as early as possible. Because the unfortunate truth is just that because these kinds of diseases like cancers are so complex, as the disease progresses and it affects all of these different networks throughout the body, by the time, if we were able to cure even that original disease, there's so many other networks that are perturbed that curing the original disease doesn't really solve the problem because now there's a whole string of other issues. So it really comes down to being able to identify individuals at high risk so that we can diagnose earlier when we start to deviate from a healthy state. And finally, I think just continuing the ongoing quest to better understand and elucidate the biological mechanisms that underlie the onset and the development and the progression of cancers. Certainly that's what we've been working on for a long time, and I think that this systems approach that we're advocating for is certainly one way that we can move forward in that area to better understand these mechanisms, because if we understand the mechanisms and we understand how it works, kind of going back to that Richard Feynman quote, then we can start to engineer solutions and viable therapies and treatment options.